I'm a neonatologist, and I tend to have a short attention span myself, so my talk is going to be a little bit on the brief side, so hopefully I won't compromise any clarity, and I'll try to be as clear as possible and hit the key points on what we've been trying to do in our research models. So, as was mentioned, I help lead the program in developmental origins of disease at the University of Iowa. And for quite some time now, we've been trying to leverage some larger animal models and figure out the environmental contribution to adult cardiovascular diseases. And we kind of go back and forth between sheep and mice and pigs, and more recently, starting to translate to humans. And it's been a lot of fun all the way through. Adam and Sean actually hit a lot of the background that I didn't need to cover in my presentation. People have been talking about the Barker hypothesis, fetal origins of adult disease, developmental origins of disease. It goes by a variety of different names, but basically for 3 decades or more, epidemiological studies have suggested that early life events, including adverse intrauterine exposures, can program the development of adult cardiovascular diseases. Most people focus on hypertension. I'm going to talk about hypertension just very briefly, and then I'm going to talk about the reason that we're really using large animal models to push forward our understanding of the origins of coronary artery disease, which is a much more difficult area to investigate, certainly in smaller animal models. So just to cover some of the initial epidemiological data, this is a study 20 years ago. Janet Rich Edwards, who's at Boston, has moved on now to her Project Viva, followed up with these American nurses. There are 70,000 nurses in their cohort, and they had self-reported birth weights, which aren't ideal, but they showed a very strong inverse relationship between birth weight and the relative risk of coronary artery disease and stroke in adulthood. And I cut this slide down to just a few different species. Hit pigs here, and this group at the UK showed what many people have shown in countless number of species now, that as birth weight decreases, your systolic blood pressure or mean arterial pressure in adulthood increases in a very linear distribution. There's no real cut point. It's not like individuals that are growth restricted at less than the 10th percentile are the only ones that suffer from hypertension. It's real in a continuum, and even just a few millimeters rise in blood pressure can lead to substantial public health implications. So it's a major problem, not just for those 10 percentilers. And to cover just very briefly the work that we've been doing in mice. As was mentioned, mice are limited for a variety of reasons, especially their size, but then also the fact that they tend to be born at a very developmentally immature time point. So you can't really try to say IUGR mouse models IUGR human. And we've shown that, other people have shown that. So we've really, in our studies, pushed the growth restriction window out to the time of weaning, at least out 14 days after delivery, if not 21 days after delivery. And we perpetuate the growth restriction phenotype by identifying mice that are growth restricted at birth, having birth weights less than the 10th percentile. Then we put them into larger litters of pups, so there's more competition for maternal milk supply, maybe more stress on them, and they are then growth restricted. So these weanling mice that were born small and stayed small by virtue of being in a larger litter end up with higher blood pressures into adulthood. This is an example of our data in females, looking at control and growth-restricted mice that have recovered from ovarectomies. These are looking at blood pressures by carotid radial telemetry when the mice are allowed to recover for a week, and then they scurry about their cage, which I agree is a very abnormal environment for a mouse to be living in, but it's the lab environment that they're born to. So these mice are allowed to roam about their cage. They resume a normal circadian rhythm, so mice are nocturnal. So 6 p.m. here, they tend to be awake and stay awake until the lights go out about 6 in the morning, or the lights turn on, at 6 in the morning, and then they tend to be a little bit more restful until they start to get ready to wake up when lights turn off again. As you can see, the blood pressures of the growth-restricted mice tend to be higher than they are in the control mice. Most impressively, they have this, what's called a non-dipping hypertension, so their blood pressures don't go down as much as they should when they're at rest. And that's a marker for increased cardiovascular risk. But even more than that is this morning surge hypertension or rising surge hypertension, where they're either anticipating arousal or they're starting to arise and their blood pressures start to spike up, which is part of the reason why adult cardiac events tend to happen, first thing in the morning or just before arising. So this is a major issue that we're looking to translate to human studies with Holter monitoring and other non-invasive techniques. So those are females. Over on this side are males. And the males are also hypertensive. So this is mean blood pressure as a percent of baseline. This is when they received artificial cerebrospinal fluid into a catheter placed into their brain. And so that's just baseline conditions. Growth-restricted mice are hypertensive. When we gave the growth-restricted mice leptin during this phase of growth restriction, we actually could prevent this adult hypertension. We came to do that because we knew that these growth-restricted mice are very leptin deficient by virtue of the fact that they're not getting leptin from breast milk, they're not getting leptin from their own endogenous adipose tissue because they're growth restricted and just don't have that adipose tissue to produce the leptin. So we can recapitulate or recover their leptin levels and then normalize their blood pressures into adulthood. The other thing this slide is showing in brevity is the fact that some of this hypertension we think is driven by central renin-angiotensin system activation feeding through the sympathetic nervous system, and we can drop the blood pressures down by infusing angiotensin receptor antagonist losartan into the brain. So this is just kind of highlighting some of the directions that we're going with our mouse models, trying to figure out why it is that they have a certain time course to their blood pressure and what components of the renin-angiotensin system are overactivated in these growth-restricted mice. Translationally, we're moving to give leptin to humans and try to prevent the developmental origins of hypertension. And really, the direction that our translational studies are going, given the fact that these are mice that are born in an immature state, we really feel like this model is more translational to the premature population I take care of clinically. Our premature babies are born obviously early. They separate from the placenta prematurely. They don't have transplacental leptin provision. They don't have placental leptin production, and they also are growth restricted, as Sean mentioned. And so we have a lot of data now that premature babies are highly and persistently leptin deficient, and we're hoping that we can correct that with some nutritional interventions and maybe help them have a little bit better blood pressures into adulthood. But going back to kind of the purpose of the talk here is to highlight some of the limitations of the smaller animal models and then highlight, more importantly, the advantages of the larger animal species. As I mentioned, unlike hypertension, there's a paucity of literature looking at programmed coronary dysfunction, and coronary arteries are very different than other vascular beds, and I highlighted just a couple examples there. Coronary arteries constrict to acetylcholine. It's not just sheep that I'm going to show you, but also human coronary arteries constrict to acetylcholine, which is more classically an endothelial-dependent vasodilator in other vascular beds. And coronary arteries are also more dependent on vasodilatory prostaglandins than other vascular beds tend to be. So in our sheep model, we wanted to play up what Adam was talking about, is that probably a major component of the developmental origins is maternal fetal stress. And birth weight in and of itself might not be the driving factor. It might just be one of the more easily measured surrogates for a threatened intrauterine environment. So we really feel fetal maternal stress is at least a major, if not primary, etiology for the downstream effects that we see. It's known in humans as well as rodents that if you inactivate placental 11 beta HSD type 2, the enzyme that metabolizes cortisol, then extra cortisol can cross the placenta, expose the fetus. It's true in neonatology that antenatal steroids facilitate maturation, helps our babies survive when they're born at a very premature time point, but it's also known if you give repetitive antenatal steroid doses, the fetus will become growth restricted. And it's kind of a dogma that the fetus can't both mature and grow at the same time. And so this whole stress, steroid exposure, we think is causing early maturation at the expense of growth. That makes sense if your goal is to just survive to the point of reproduction, when biologically you become less relevant. But nowadays we're able to keep people going for longer, and then they start to have some of the detrimental effects with hypertension and coronary artery disease. So this is our model that we've been using. We give dexamethasone, and the reason that we can give dexamethasone is because it bypasses this 11-beta-HSD enzyme and goes direct to the fetus. So sorry, this is the model that we're using. We're giving dexamethasone 0.28 milligram per kilogram per day to the pregnant ewes pretty early in gestation, 27 to 28 days gestation, with term being 145. And then we take control and dex-exposed offspring and study them shortly after birth, and then again in kind of an adolescent time point. And what we noticed was that the sheep that were exposed to dexamethasone tend to have slightly higher blood pressures when they're newborns, like a week old, and then they have significantly increased blood pressures in adolescence. And that was kind of fortuitous for us because we really wanted to figure out the time course of what we were interested in. And it's better if we can look at a window when the sheep are hypertensive and see if any vascular changes might be related to that. More importantly, if those vascular changes are also present even before the onset of significant hypertension, it's kind of a chicken and egg thing, and it helps us figure out if there's a primary vascular pathology or if any of the vascular changes we were seeing are just due to the blood pressure itself. Here again is just highlighting how unique the coronary arteries are. So I have three different blood vessels here. I have carotid arteries, which are in the blue, the coronaries are in the black, and mesenteric arteries are in the red. The thicker lines are the steroid-exposed arteries, and this is looking at responsiveness to angiotensin II. So the mesenteric arteries respond very little to angiotensin II compared to their response to potassium chloride. The carotid arteries have a fairly strong response, but it's only at higher concentrations. And then the coronary arteries respond at fairly low concentrations and respond almost to the same level that they did the potassium depolarization. And you can see here the dex-exposed coronary arteries were hypercontractile compared to the control coronary arteries. And then looking at mechanisms, then we knew from studies that were ongoing that one of the major downstream mediators of angiotensin-induced hypertension or vasoconstriction was superoxide production. So we're looking at superoxide production by lucigenin-enhanced chemiluminescence. This is on a scale of relative light units produced per milligram vessel per second. And you can see the dex-exposed vessels, even at baseline, produced almost twice as much superoxide as the control vessels did. And then when we incubated them with increasing concentrations of angiotensin, you can see with large error bars again, because sheep tend to be a lot more variable and have a lot smaller numbers than you do on mice. But still, even despite the fact that the baseline was higher in the DEX group, they also had an augmented superoxide response to angiotensin. And we wanted to look at that by a couple different assays. So here we're looking at superoxide detection also by dihydroethidium fluorescence, confocal microscopy. And so this is a DEX-exposed vessel that didn't get any angiotensin. The endothelium is faintly lined in white, which is detecting the dihydroethidium. The hydroethidium gets oxidized by superoxide and intercalates with the DNA, and then we can kind of see it fluoresce here. The control vessel didn't look too different from the DEX-exposed. Control with angiotensin incubation on the bottom right here likewise didn't really have much of a response. And we thought I mean, this is representative of 3 or 4 coronary arteries, maybe, that there was a suggestion of increased superoxide in the endothelium of the DEX-exposed vessels. And it was at least consistent with what we had seen by the mucigenin-enhanced chemoluminescence assay. So we're kind of pursuing that. And then at the same time, one of the other advantages of sheep is the fact that you get so much coronary artery that you can look at a variety of different things from the same animal. So while it might be difficult to acquire the sheep, once you get them in hand, then you can look at a variety of different things side by side. I showed the picture earlier of the muscle bath. You can take 8 different coronary artery segments and incubate them with a variety of different conditions and really try to figure out what mediators are important. In here now we're looking at the prostaglandin pathway. We're placing indomethacin in the muscle bath, which is a cyclooxygenase inhibitor, so it's inhibiting breakdown of arachidonic acid to vasodilatory, vasoconstrictor prostaglandins. We're trying to constrict the coronary arteries with thromboxane mimetic U46619. And you can see the DEX-exposed coronary arteries are hypercontractile to the thromboxane mimetic. And contractions for both the DEX-exposed and the control vessel go up fairly dramatically and reach the same level when we inhibit prostaglandins. So our thought there was potentially the DEX exposure was leading to an underproduction of vasodilatory prostaglandins, and with the cyclooxygenase inhibition, they kind of both come up to the same level then. And to try to figure that out in a more accurate way, we went ahead and cultured vascular smooth muscle cells from coronary arteries. This is just an example of what we were seeing. And then we incubated them with angiotensin for about 4 hours, and then ran them through a HPLC system and looked at the percent of arachidonic acid, that radiolabeled arachidonic acid that was metabolized. And so for control cells, they metabolized about 10% of the arachidonate. DEX-exposed cells only metabolized about 3%. And the thing that they were deficient in was really PGE2, which people, cardiologists and neonatologists will know that's the medication that we infuse into babies. I think someone was talking about it with their ECMO study to try to maintain patency of the ductus arteriosus. So it's a major vasodilatory prostaglandin that these coronary muscle cells aren't producing. This other analyte that we detected at fairly high levels was the stable metabolite of prostacyclin. That didn't seem to be as affected. So to kind of conclude a little bit this first example in sheep, we feel like the inciting antenatal event, in this case excessive glucocorticoid exposure, is really throwing off the balance of vasoconstrictor, mediators, superoxide, vasodilatory components, prostaglandins, and leading to an imbalance of constriction and dilation, leading to a primary coronary artery dysfunction, again, in the absence of significant hypertension. To move quickly into the second example, so we got known a little bit on campus for doing cardiovascular research, vessel research. The pulmonologists were coming to us and saying, hey, we think that a lot of our cystic fibrosis patients are having higher blood pressures. Do you think they could have a vascular dysfunction as a component to that? And we kind of launched into it head first, and then several months later we kind of looked at the literature a little bit and actually saw that maybe their blood pressures would tend to be a little bit lower than they would be in other populations. But it was exciting time to kind of get involved in it. Cystic fibrosis is a major problem. It's the most common life-limiting autosomal recessive disease in people of European heritage. The delta 508 mutation is the most common cause of cystic fibrosis, and it has a very high carrier rate, suggesting that carriers might have some advantage, a heterozygote advantage. Most likely explanation for that is that it decreases salt losses with diarrheal illnesses such as cholera. But we were wondering if there was maybe a protective cardiovascular phenotype as well. The reason we were wondering about cardiovascular protection is because CFTR delta 508, This mutation leads to misfolded protein that gets retained in the endoplasmic reticulum before it's targeted for early degradation. So it doesn't reach the cell surface. Individuals then have a loss of function, leading to lung disease, pancreatitis. But then we were wondering if the short time that it's having increased residency in the endoplasmic or sarcoplasmic reticulum, whether that could lead to a gain of function at the level of calcium release. Again, once we got looking into the literature a little bit more, it was known for quite some time, back into the 70s really, that young adults with cystic fibrosis tend to have lower blood pressures, but everybody thought it was just because they were hypovolemic or sodium deficient, and like the salt wasting leading to relative hypovolemia was the cause of their relative hypotension. But even as early as 2001, it was known that they had increased vasodilation to sodium nitroprusside. And again, the question was whether retained CFTR could be decreasing vascular tone. There was a prominent publication in Hypertension looking at delta 508 carrier females, and they had lower pressures as well. But there again, the carriers with the lower blood pressures tended to be the ones that were wasting the most chloride in their sweat. So there was still correlations between CFTR activation and blood pressure that were difficult to disentangle in human populations. The weakest form of evidence was from this Framingham Heart study, which is a GWAS study, and the only SNP in that study that associated with flow-mediated vasodilation was in CFTR. So take that for what it's worth, but a highly significant result in their study. So we wanted to figure this out. We started with the mice because the mice give us the best opportunity to do longitudinal blood pressure measurements. And it also opened the door to measure blood volume in carriers of the delta 508 mutation. Some of my colleagues were doing transfusion medicine and had developed a biotinylation assay where you can biotinylate red cells, transfuse them, and then resample later and determine blood volume by flow. And so when we looked at wild type and delta 508 heterozygote mice, we noticed that blood volume was actually no different, if anything, a little bit higher in the het mice. Looking at blood pressure, consistent with what was reported in all the human studies, delta 508 mice had lower blood pressures here pretty much throughout the day. I'd have to note here, we had intended to look at CF mice, homozygous delta 508 mice, but despite the fact that they were on a special diet with a special form of bedding, they had about a 30% perinatal mortality rate, and we couldn't get any of them to survive surgery, really. So they are pretty sick. The Murine CF model is heavily criticized because they don't have lung disease. We thought that was to our advantage. If we can separate the lung disease and the cardiovascular disease, then maybe we can figure out again if there's a primary cardiovascular phenotype. So it wasn't bad for us that they didn't really have lung pathology, but it does a little bit invalidate the whole premise of studying CF in mice. But we did notice, the blood volumes were retained, but their blood pressures were still low. We looked at aortic responsiveness to noradrenaline and saw that the het mice didn't constrict as well to noradrenaline. So we wanted to move again into a larger animal species so we could really look at some second messengers. And we were able to take advantage of the fact that there was an active CF research group. They have a CF pig model that they've been making some great strides with. And so we were able to get some aortas from them and then test our hypothesis that these CFTR null piglets would have a loss of CR charge and neutralization and decreased cytoplasmic calcium. I have a cartoon here. One of the journal editors asked us to try to make sense of some of the different pathways that we were trying to interrogate with our rig. But CFTR is highly expressed in the sarcoplasmic reticulum membrane, and we thought it could be charge neutralizing the flux of calcium. And we also thought that it could be contributing to potassium chloride responsiveness at the plasma membrane level because voltage-dependent calcium channel release could be inhibited if the cell membrane was hyperpolarized. So here's some of our hypotheses based a little bit on known function of some of the different channels. So just very briefly, we published this now in Journal of Cystic Fibrosis a few years back. This is newborn pig vascular smooth muscle cells. Consistent with the hypothesis, vessels from null piglets had decreased intracellular calcium levels at baseline. This is an ion optics rig that we're using to measure this. And mice that were heterozygote or homozygous for the delta F508 mutation tended to have fairly normal baseline calcium levels, but then relatively low stimulated levels. So if you look at the ratio of the two, like a fold change in response to angiotensin stimulation, the delta F hets and the homozygote smooth muscle cells tended to be less calcium responsive. And that translated then to changes in aortic tone. I presented this to the CF group, Mike Welsh and others. They weren't sure what to make of it. They had historically been including hets into their wild type group just to kind of increase their numbers because they thought they were non-CF animals. But I told them I'm not so sure I would jump to including hets with your wild type because I think there's probably 4 different groups: wild type, null, het, and delta F homozygous. I'm not showing hets here because they tended to not give me as many hets going forward. So I'm showing the delta F versus the null and the wild type, and in particular the delta F tended to be very under-responsive. So in conclusion, we showed in adult mice perinatal growth restriction increases systolic blood pressure, and the delta F508 mutation decreases systolic blood pressure. But we really weren't able to move very far past blood pressure phenotypes in the mice. So I kind of leveraged things and moved into sheep to look at coronary arteries and then also look at the time course for some of the changes that we were seeing. So sheep coronary arteries had increased tone, increased angiotensin-stimulated superoxide production, and decreased prostaglandin production. Whereas with the delta F mutation, there's decreased aortic tone and decreased calcium release. So we really feel like the models are very complementary to each other, and we, to this day, don't really want to be using a single source for our investigations. We've enjoyed using sheep. You can do a very precise defined exposure. Again, the large size allows for investigations early in life. We've also done some of the intrauterine surgeries and measured blood pressure in intrauterine animals with exteriorized uteruses. And you can collect neonatal coronary arteries in sheep, which you can't do in mice. The porcine models have very similar advantages to the ovine models. Transgenic ones are online. I borrowed this from Randy's website. Murine models can be disadvantaged by the fact that maturation can occur after delivery, but then we're trying to turn that into an advantage by using it as a model then of prematurity, more so than intrauterine growth restriction. And then we've also started to do fish models, actually. All of these things that are considered weaknesses could likewise be considered strengths. The fact that fish develop apart from a placenta is actually very advantageous from some of our studies where we're looking at antidepressant exposure. Antidepressants affect the mom, affect the placenta, affect the fetus, and we're trying to figure out if there's a primary fetal effect of the exposure. There's really no way to definitively do that unless you separate the embryo from the placenta and the mom. And zebrafish have transparent embryos, and you can kind of see the little heart. Sorry if I'm not pointing at the screen you're looking at, but the little heart is surrounded by a pericardial window, and we're seeing pericardial edema and changes in looping patterns in some of these models. So I think every species has something that we can learn from it. For the large animals, challenges do exist. We've had difficulty with procurement at times because we're not always funded to primarily do large animal research. I think longitudinal assessments could be challenging and taxing on the system, and genetic manipulation is starting to come online, and I think that's going to pretty soon no longer be an insurmountable or even a significant challenge. Again, collaboration's critical, and I think there's a lot of future In neonatology, I think sheep have been a tremendous species because it was part of the reason that we know to give antenatal steroids in threatened preterm labor. It's part of the reason we know to give surfactant. And I know a lot of investigations are being funded looking at transitional physiology and trying to figure out better ways to take care of babies, because I think neonatal sheep are one of our best models for prematurity. And so with that, I'd just like to thank everybody for their attention, thank my collaborators, some of whom are pictured, the ones in bold, and then mention the fact that we just completed construction of our new children's hospital, which is right next to Kinnick Stadium. And some of our babies have moved in, some of our pediatric patients have moved in, and the surgical patients will be moving over shortly. So hopefully everybody will have a chance to visit us there at some point. So thank you very much for your attention.
Any questions? I'll start with one. Have you checked if your cardiomyocytes from the dexamethasone exposed sheep have any different responses to prostaglandin, or have you tried to interrogate that aside from your aorta?
Yeah, for the cultured cardiac myocytes, we haven't really done a lot of phenotyping of those. We looked at the coronary smooth muscle cells, but not so much the cardiac myocytes. I think that would be interesting to look at.
So this is just, I don't know if it's worth it or not, but you can take it. There is a mouse model of cystic fibrosis that you might want to look at it. It's a transgenic one that overexpresses beta ENaC. And that has all the features of cystic fibrosis in the lung. I have no idea what it does in the cardiovascular system, but as the CFTR is affected in both mice and in humans, then the beta epithelial sodium channel goes up and overexpressed in this group. And Rick Boucher at North Carolina and Marcus Mall did that.
I think that's great. I think all these different mutations, we can learn something from them. The medical treatment of cystic fibrosis is changing rapidly with the medications that can facilitate movement of the CFTR channels to the cell surface and everything. So I think looking at the different mutations can have a lot of advantages. And I'm sorry if I seem to suggest that there was any limitations with any of the different models, because as I was trying to highlight as much as anything, I think every single model, every single mutation, you can learn something from it. And the key is kind of integrating it and trying to figure something out that makes sense, not just for that species of genotype of mouse, but for individuals, humans especially. So.
Well, thank you so much, and let's thank all of our 3 speakers from the session.